Transcription consists of 3 steps: initiation, elongation, and termination. On the DNA strand, there is a promoter sequence that tells the RNA polymerase where to bind. The RNA polymerase recognizes this area because of a region called the Tata box, which contains a series of thymine-adenine hydrogen bonds, which are easier to break than the cytosine-guanine bonds. When the polymerase binds, a replication fork is created. Next, elongation occurs. This is where the RNA polymerase begins adding base pairs, but keep in mind that it only adds nucleotides to the 3' end. Note that the RNA polymerase binds on the template strand of DNA and that the coding strand, despite its name, actually does not get transcribed from the coding strand as non-template. Traditional base pairing rules apply, except in RNA, the thymine that normally bonds with adenine in DNA is replaced with RNase uracil. Finally, termination occurs. Eventually, RNA polymerase will have created a strand of mRNA from the template DNA strand and reach a region in the DNA called a terminator. As can be guessed from the name, the terminator tells the mRNA to detach from the DNA, where it will then begin working on some other transcription project in the cell if needed. Translation and mRNA splicing follow. The importance of the 5 and 3 ends of the DNA can be seen especially in the elongation phase of transcription. These numbers are given through the names of the carbons in the DNA and RNA structure, and the 5' end is always closer to the phosphate group in DNA. During this phase, RNA polymerase adds base pairs to the 3 ends of DNA to form pre-mRNA. The coding strand is from 5 to 3, while the template strand moves from 3 to 5. The direction of these strands is necessary for transcription to occur successfully.
The translation follows the same process as transcription in that it has 3 steps: initiation, elongation, and termination. During the initiation, the tRNA, mRNA, and the amino attach to initiate the elongation process. Before the tRNA's anticodon can bind to the start codon, the amino acid has to bind to the tRNA with aminoacyl tRNA synthetase. After this, the tRNA with the appropriate amino acid binds to the mRNA start codon with the A site of the ribosome. The second step is elongation, which is activated by GTP. GTP is used as an energy source to move the ribosome towards the 3' end of the mRNA and move the tRNA from the A site to the P site of the ribosome. Once this happens, rRNA catalyzes the formation of a peptide bond between the amino acid and the carboxyl end of the polypeptide in the P site. Elongation then removes the amino acid from the P site and attaches it to the growing polypeptide in the A site. After this, the ribosome unit moves the tRNA from the A site to the P site, and the tRNA which was in the P site is moved to the E site, the exit. This continues on until the ribosome reaches the stop codon. The last step is termination. Termination begins once the stop codon reaches the A site of the ribosome. The stop codon is recognized by a protein called the release factor, and it triggers the separation of the polypeptide chain from the tRNA. After the polypeptide chain is released, everything else falls apart and separate from each other.
RNA splicing is the modification that happens to the precursor mRNA in order to transform it to mature mRNA. During splicing, introns, non-coding segments of RNA, are cut out from the strand. The exons, coding, expressed segments of RNA, are joined together. For eukaryotic cells with introns, RNA splicing is necessary to create an mRNA molecule that can be translated into proteins. This process can be carried out through using spliceosomes. Spliceosomes are a complex of smaller ribonuclear proteins, or snRNPs, that can carry out the excision. First, a group of snRNPs is required in order to cut out the introns of the mRNA. The group where snRNP binds to the intron portion, causing it to fold inward. This folding causes the 3 and 5 of the intron closer together, forming a loop. The exons also move together. The intron detaches and the splice sites join together to form mature mRNA. The detached introns are just for other purposes, while the detached snRNPs are reused for splicing. Amino acids are extremely important to how a body generally functions. Following the transcription of an mRNA strand, the process of translation is carried out in ribosomes located in the cytoplasm for eukaryotes. In prokaryotes, the process of transcription and translation are paired, meaning that translation begins immediately after transcription. The triplet code, or codons, that the mRNA contains determines what type of amino acid should be added onto the polypeptide chain. The mRNA will continue to code for more amino acids until the appearance of a stop codon. After the termination of the mRNA sequence, as marked by the stop codon, the polypeptide chain product may still have to go through several processes. Since it is still a polypeptide chain, meaning it's in the primary structure, it still has to fold into the correct 3D shape. The primary form refers to the chain-like structure that has just been produced by the translation process. The secondary structure refers to local folded areas that form due to interactions of the atoms as the backbone. Common secondary structures are the pleated sheet and the helix. The tertiary structure refers to the overall 3D shape that has been affected by the interactions of the R groups that make up the proteins, such as hydrophobic interactions and disulfide bonds. The quaternary structure is formed by multiple polypeptide chains, known as subunits.
DNA replication is not always perfect. Sometimes mutations can occur. Point mutations are alterations of one nucleotide base pair of a gene. A nucleotide pair substitution is when one nucleotide and its partner are replaced with another pair of nucleotides. Missense mutations enable a codon to code for an amino acid, even if it's not the right one. Nonsense mutations change a regular amino acid codon into a stop one. Insertions and deletions are the additions or losses of nucleotide pairs in a gene. If they interfere with codon groupings, they can cause frameshift mutations.